Hello, I am Dr. Dan Kalish, and welcome to my class for our fall mentorship mini series. And this is on brain health, amino acid markers. Specifically, we're going to talk about amino acids. And this is, I don't know, perhaps my favorite subject. It's a very interesting subject. It's something that most people don't study that much or that carefully. And first of all, it's really interesting. And second of all, it's a super common problem. And thirdly, you can help gazillions of patients. Probably most of your patients have some kind of amino acid imbalance. Like in the last 10 or 15 years, I cannot remember a patient whose entire amino acid markers throughout the entire series of tests that we do were all normal. Like, I don't even know if that exists. So you're going to have instances where either you're going to see amino acid markers that are low on the amino acid test that we're talking about today, or you're going to see it on organic acids testing. You're going to see implied amino acid deficiencies because of oxidative stress, because of neurotransmitter imbalances. I mean, all detoxification pathways, you name it. The neurotransmitter supports 5-HTP, tyrosine, or amino acids. The detoxification support methionine, glutathione, methionine, glutamine, glycine, N-acetylcysteine, cysteine, those are amino acids. The liver support, glycine and taurine, those are amino acids. Joint support, I mean, it goes on and on, right? This is a huge subject area. What we're looking at specifically today are amino acid test results from plasma, but the amino acid treatments expand out from that quite a bit. So if you're brand new to the Kalish Institute, I want to welcome you. I am Dan Kalish. I founded the Kalish Institute 2006, trained thousands and thousands of practitioners. I think it's over 10,000 now, have gone through one or more of our courses. I've worked with Mayo Clinic. I've worked with Richard Lord, whose work we'll be talking about today. I've been in practice for over 30 years. I managed to pass the IFM certification test, which was challenging, let's say, but I passed it, and I'm IFM certified. And kind of ready to go here now for you guys in terms of what we're going to do with the protocols. All right. And so we're gonna take a look at a couple of different things. I'm gonna go through some slides, then we'll show some labs, and then I'll talk about specific protocols or programs that you can use. The reason why we call this the mentorship mini series is because this is a mini little look at a fraction of a percent of what you get if you sign up for the mentorship program. So the mentorship program is 1 year of what you're doing today for 1 hour. It's a massive course. I've been teaching it for a very long time. I'm very excited about it in general, and the offerings that we can provide to you through the class are pretty profound. And any of you that are interested in really, taking your practice to the next level, I strongly encourage you to check it out and see if it's if you're an appropriate candidate for it. Okay. And so let me just clear one thing here. So if you're at all interested in becoming an expert at lab interpretation, check this out. We start on September 16th. You get a very large bonus if you remember this code, Mentor Mini 24, $1,299 off the mentorship program. You can also just scan that QR code there. We have a few spots that are still open. It's a limited amount of people that we take each time. The fall group tends to be our most robust, rowdy and fun group. I don't know why. It's the September people are kind of like into it, So if you're an into it kind of person, sign up for the class, check it out. You can talk to one of my salespeople. You can talk to myself about the course, and it's a week away. So if you're thinking about it, jump on it and let's get you all into action so you do something. And we want you to be at your highest level of skill in interpreting these tests. It's really, in my mind, if you're not taking my program, that's fine. But if you're not taking some program where you're spending a year or two learning how to interpret labs, then, shouldn't call yourself a functional medicine doctor, in my mind. I mean, that's a little snooty, but I spent 8 years studying with Dr. Timmons and 4 with Frieder and 12 with Dr. Lord. So I'm into this for 20 years of postgraduate education. You have to put at least a year or two in. Again, it doesn't have to be my class. Just get yourself in a program where you have a mentor that can really teach you how to do these labs. And then, in a year, you'll be an excellent practitioner and you'll have that the rest of your career. So I encourage postgraduate education as a way for us to really understand what our role should be in helping patients with labs, okay? So key understandings on amino acids. And I wasn't taught this until I was in year 20. The main role of amino acids, the main role of all these amino acids that we're talking about is not in their individual functions. The main role of all these amino acids is in their roles in protein synthesis. So there are 20 amino acids, and we need all 20 of these amino acids to make insulin, hemoglobin, every antibody, all these things. There's one sort of large exception. All 20 are not required to make collagen. Okay, you only need 3. But outside of collagen, all the proteins in the body are synthesized from 20 different amino acids. You also need amino acids, all 20 of them, to make enzymes, like the detox enzymes, the famous cytochrome P450, pyruvate dehydrogenase, which is the one that's involved in insulin resistance, lactate dehydrogenase, also involved in metabolic function, succinate dehydrogenase, involved in everything to do with energy production. All these proteins in the body can only be made by 20 amino acids. So the key role of any individual of those 20 amino acids is in protein synthesis. Protein synthesis. The non-protein functions, let's take a common one like tryptophan. So when I say tryptophan, you are immediately already thinking about serotonin, okay? Oh, sorry, I didn't start my slides up again. You're already immediately thinking about serotonin, right? And so in the world of clinical nutrition, we tend to pigeonhole an amino acid into a certain role. So tryptophan, you think serotonin. What's another one? Glutamine, everyone's already thinking leaky gut repair. Tyrosine, you're thinking either thyroid or dopamine. But these amino acids, the 20 key amino acids, have another role. Their non-protein function might be like glutamine helps repair the gut lining. But the protein function is that you need all these 20 amino acids, including glutamine, in order to make proteins. All proteins in the human body require the same 20 amino acids. So in addition to their individual roles that they play, like tryptophan makes serotonin, tryptophan is also one of the 20 that's essential, that's required for making all proteins in the body, except collagen is the exception. Right? The single carbon pool is another example. That's how we produce, that's how we go through the process of methylation. That relies upon amino acids. You need 3 amino acids to make glutathione, cysteine, glycine, and glutamine. You need about 6 or 8 of them to run your phase 2 detox pathways. You need amino acids like arginine and citrulline to help regulate blood pressure and cardiovascular health. You need glutamine, as I mentioned, for gut health. So there's a whole bunch of reasons why we get into amino acids, but where it gets a little slippery is in not understanding the protein synthesis production function of amino acids. I mean, in fact, I didn't really ever think about this until, again, I was in practice for over 20 years. And one of the first things that Dr. Lord did when he was training me was show me how to interpret these amino acid tests and explain to me why they are so important. It never even occurred to me that there was anything except the non-protein roles of amino acids. So in clinical nutrition, the way that amino acids are taught is tryptophans for serotonin, glutamines for your gut. Nobody's standing up and saying, wait a minute, the main role, the main purpose of these amino acids is protein synthesis. Which proteins? All proteins in the human body except for collagen require the 20 amino acids. When you're doing the tests, it's much preferred that you do plasma amino acid levels, not urine. Urine amino acids show you what you're kicking out. Plasma shows what's circulating in the body. And the plasma amino acids are a measure of a combination of obviously what's coming in and what's going out of the blood. These are the 20. These are the 20, and they're listed in their order of amount that they tend to occur. This is crazy. I didn't believe this when Dr. Lord first taught me that. I was like, is this even possible? Like, how was I in practice for 20 years and didn't know this? Every single one of these 20 amino acids is required to make every protein in your body, except for collagen. And get this, in almost the exact same ratios. So like if you're making a cytochrome P450 enzyme, or you're making some insulin, or you're making, I don't know, some protein that's in your heart, you're using the same exact amino acids in about the same ratio. So why are all these proteins so different? It's because of your DNA sequences them differently, puts them in a different order. But it's not different amino acids. It's the same ones over and over. And the one that's the most vulnerable is tryptophan. You see tryptophan's at the bottom of the list here? It's at the bottom of the list because out of all the 20 amino acids, it's the one that shows up in the smallest amount in all the proteins in your body. Okay, so if there's an even small fluctuation of tryptophan, you got a really big problem, because remember, you need all 20 of these to synthesize all proteins in the body. So a tryptophan deficiency on a lab, you're going to think serotonin. That is the least of that person's problems. Their major problem is that protein synthesis has been interrupted, and that's what we call healing, right? That's where the repair process of the body now has been, reduced dramatically. I don't care how much protein you're eating all the time. If you run a test and a person's consistently low in any one of these 20, let's say they're low in glycine, you still can't synthesize proteins. You could be eating hamburgers and turkey and protein shakes all day long. It's not going to have the effect that you intend if you don't have the specific aminos that you need for protein synthesis. So another kind of a reverse engineering way of saying this is a plasma amino acid test is one of the most, if not the most fundamental tests that you can run. And understanding what this test means and how to interpret it and how to fix these amino acid deficiencies is critical. And remember, we're not talking about specific non-protein functions of amino acids. In that first whole section of this little lecture here, we're just talking about the ability to make proteins, and that is the ability of the body to heal and repair. Now, total change of subject. Now we're going to talk about specific things that amino acids do outside of synthesizing proteins. So we just went from the most important thing in the body, which is being able to repair with these 20 amino acids. I should just stop the lecture right now. This is the most important thing here, is these 20 and getting these in balance so that the body can make proteins. Now we're getting into the weeds on specific amino acids, and this is what in clinical nutrition people tend to focus on obsessively, and they forget about this part, which is the most important part. So don't forget about this part. That's why I started there. Now, you also use amino acids, for example, to make glutathione. Cysteine, glycine, and glutamine are all required to make glutathione. And that's the body's most important antioxidant. So that's pretty important. So if you're low in glycine, for example, is glycine on this list here? Yeah, it is. So if you're low in glycine, the biggest problem that that patient has is that they don't have one of the 20 amino acids to synthesize proteins. The next big problem they have is they don't have enough of the right amino acids to make glutathione. Is cysteine on this list? Yeah, it is. It's right there. Is glutamine on this list? Yeah, it is right there. So glycine, glutamine, and cysteine are the 3 amino acids you use to make glutathione. So if one of those is low, or they're all 3 low, most especially if cysteine is low, you suspect there could be some low glutathione problem. However, that's the least of the problem. The bigger problem is that they can't synthesize proteins because cysteine is one of these 20. Hopefully this is starting to make sense. So there's the non-protein functions of amino acids, and then the protein synthesis functions. And again, here are the 3 that makes glutathione. Another sideline for why amino acids are so important is right here. In order to conduct methylation, you need to have something to methylate. You need to have some carbons, and the carbon pool, the single carbon pool, we draw our carbons from amino acids: serine, glycine, histidine, glutamine, and tryptophan. Again, so we're back to picking on tryptophan. If you're low in tryptophan, you're not going to be able to, well, maybe there's 5 of them. Let's say if you're low in serine, glycine, and tryptophan, maybe 3 of these 5, or all 5 of them, you're not going to be able to methylate properly because you're not going to have the carbons that you need to feed the single carbon pool. Okay, you could probably be low in 1 or 2 of these and get away with it, but not the bulk, not the majority of them. Sulfur amino acids required for detoxification. It's the exact same problem. Here we have phase 2 detoxification, sulfation, glucuronidation. All these are run by nutrients like methionine, cysteine, glutamine, glycine, taurine, etc. So again, we go back to our primary list here. If you're low in glycine, glutamine, and cysteine, you might not be able to run detox pathways. If you have a significant problem with methionine, there's a whole host of issues you're going to have with detoxification and methylation. But the bigger problem with low methionine, the biggest problem is that you cannot build proteins. Which proteins? All proteins in the body. All proteins in the body. And just if you only just think about antibodies, think about all these people with long COVID or COVID reactions or weakened immune systems, just for the reason that you need to make antibodies alone, which are all proteins, right? You have to have methionine present or it's not going to work. So specific functions of amino acids, detox, general, bigger picture, protein synthesis. When Richard explained this to me, maybe this was 12 or 14 years ago, I didn't believe him at first. Then I didn't understand him. And then I started to treat patients as he was indicating I should. And then I couldn't believe the results that I was getting from that. So this is not like a little clinical trick like, oh, you know, I learned a new tool today. This is kind of cool. I'm going to do this once in a while. This is every single human being has to have all these amino acids present or they can't do any kind of healing or repair. It's just not possible. This is like a fundamental aspect of cellular repair and healing is to be able to synthesize proteins. Okay, so somebody out there is going to send me an email later and say, well, what about cell membranes? Yeah, cell membranes are, you know, I would maybe equally important. You have to have cell membrane function too. So you also want to test fatty acids and fix cell membranes. Get protein synthesis going, you get cell membranes working, you got a healthy patient on your hands. Again, this is saying what I've been saying for the last 12 minutes here, 20 minutes. Any single amino acid low of the standard 20 amino acids will limit all protein synthesis in all sentient beings. Oh yeah, it's not limited, right, to human beings. This is why we want to prescribe amino acids properly. So now, non-protein functions of amino acids. This is what we kind of get hung up on most of the time. So we don't want to obsess on this, but I just want to mention it, you know, so you can get, okay, methionine and cysteine are really important for detoxification. Lysine is a precursor to carnitine. Tryptophan, we talked about ad nauseum already, is a precursor to serotonin, okay, and so on and so on. So those are the specific functions, and you basically need to learn both sides of this. You need to learn the protein part and how you're going to help with the synthesis of all proteins, and then you need to learn the non-protein part. Okay, does that make sense? Now, once again, this is a little reminder here. Why is this so important, the non-protein functions? I mean, the protein functions, right? Digestive enzymes are proteins. We have transport proteins like hemoglobin and albumin. We have structural proteins that hold the body together. We have some hormones that are proteins like insulin and thyroid hormone. Defense, I mentioned already, immunoglobulins, antibodies, that kind of stuff. Contractile proteins that move us around. Okay, so protein synthesis covers huge amounts of the things that our patients are suffering from when you think about it. So if you think about patients who don't digest well, who have a lot of structural pain, who have hormone or insulin-related problems, thyroid hormone or insulin-related problems, all this is going to come right back to protein synthesis. Now I'm going to skip through a few of these slides and let's see here. Yeah, so we're gonna focus on. I'm just going to pause for a moment and let everybody take this in, because now we're going to shift gears and we're going to get into the non-protein functions of amino acids, okay? So now, I want to try to tie together how these are related, and then we're going to get into one very important aspect of amino acids, which is the brain, and then we're going to look at labs and design some programs. So as an example here, if you run low on glycine because you have some kind of a toxin issue, let's say that you have an exposure to glyphosate, or that you have a mercury fillings in your mouth, or the patient has some kind of environmental toxin exposure, which is pretty common. Maybe they have like a mold related problem, or even could be internally generated, like a gut related problem, something that's generating a lot of toxins in the system that they have to deal with. That can deplete your glycine. Now, once your glycine's low, because of this overwhelmed detox pathway that's been working so hard, you're also not going to be able to make proteins, all proteins. What if you're stressed out of your mind, going through a divorce, you have a financial problem? I don't think this could ever happen, but maybe there's like a worldwide pandemic and everyone's really stressed for a while. Something like that goes on, and your catecholamines just take a hit. And so your tyrosine levels drop because you're making so much adrenaline, noradrenaline, epinephrine, whatever you want to call it, right? You're making all these catecholamines all the time. So your tyrosine levels just drop. That's going to have an impact on this bigger picture of protein synthesis, okay? And so this is what we see in our patients, is they have a reason why a single amino acid dropped. And if it's one of those 20, then that's going to have a global effect. So the protein synthesis functions of amino acids, the larger non-protein functions, and now we can kind of drill down specifically to just brain-related problems. So, and then when we look at the labs, I'm going to try to tie this all together. So when you're looking at neurons being damaged and not, the brain not functioning quite well, it can be due to low amino acids, which would lead to low neurotransmitters. It could also be damage to the brain cells themselves. And it matters, right? Because if there's just a deficiency state and you see low amino acids, you restore the amino acid levels and then things balance out. If the problem is that the brain cells are damaged, it may be that your tyrosine levels are fine, but that the body needs more than the average amount of, let's say, dopamine to function well. So you're going to see some contradictory results on these labs, just to warn you. You might see a dopamine marker that's present with low tyrosine. You might see a dopamine marker that's present with normal tyrosine. These are independent variables to a certain extent, although they of course overlap. If I have a mouthful of mercury fillings and I eat tuna 3 times a day, every day. Actually, I have a couple that's like this. It's a married couple, and they own a chain of fish restaurants. You couldn't make up half of these stories if you wanted to. And they've owned this chain of fish restaurants for like 10 or 15 years, and there's like 5 or 6 fish restaurants that they have in some area on the East Coast. And so they eat fish in their fish restaurant because they work obsessively and eat at the restaurant like a couple times a day for all this time. So if you eat enough fish, you can definitely become mercury toxic. But most of the time, it's an amalgamation or an accumulation of environmental toxins from a whole bunch of different areas. If that starts to accumulate enough and there's damage to the brain cells, a normal amount of dopamine might not get those damaged brain cells to work really well. You can have the same exact problem with not enough dopamine if simply you don't have enough neurotransmitters being produced because your tyrosine is low. But when you start to look at these labs, it's going to be confusing. You might see a serotonin metabolite marker that is problematic, but the tryptophan levels are fine. You might see low tryptophan, but the serotonin metabolite markers are off. So as we look at these labs, there's going to be some what seem like things that shouldn't exist at the same time together. I'll try to explain as we see the tests why that's the case. But just so you know, it's not inconsistencies in the lab. It's because the markers that we use, for example, to measure serotonin or dopamine are looking at metabolites. And now we're also talking tonight primarily about actual amino acids. And you kind of have to learn these separately, and they're not always going to correlate. It's not as hard as it sounds, though. If you see a low amino acid, you're always going to treat it. But you may use that same amino acid even in a case where the amino acid levels are not low. Does that make sense? Anyways, when you see the labs, hopefully that'll make sense. So if you have a damaged neuron, the amount of dopamine you may need to get it to function well might be larger than the average person. You might also see some excessive metabolism or production of the dopamine, even in the presence of normal tyrosine. The more damaged the neuron is, the more this discrepancy is going to show up. In a non-damaged system, if the person's low in tyrosine, you give them a little tyrosine, they're going to get better in a couple of days or a couple of weeks. In a system where there's neuron damage, that same amount of tyrosine might not have the effect that you intend as quickly as you would think, or you might need the dosages to go up quite a bit. Okay? And this is kind of an example if you're thinking about there could be enough gas in the tank, you could have enough tyrosine in the system or even enough dopamine in the system, but if it's not, engine's not running right, you've got a deeper problem there. Or it's like another example I use with patients sometimes is like if you have a garden hose and it gets a kink in it or you step on it, then the flow is not working well. So there's many reasons why damaged neurons are going to interfere with neurotransmission even when the amino acid levels are normal. Okay, so let's take a look at some labs and try to make some sense of this. And then towards the end, we'll go over questions, okay? So if you do have questions, type them in there, and I'll come back and try to get as many of the questions answered as possible. If you came late, this is the Mentorship Miniseries, which means that we're starting a mentorship class next week. You can scan that QR code and sign up that way. You can enter this code, MentorMini24, to get a rather substantial $1,299 discount. This is a one-year training program where you are reviewing labs with me and a small group of practitioners every week live, your labs, your colleagues' labs. We're going after case after case after case after case. We have medical doctors, nurse practitioners, chiropractors, acupuncturists, a lot of dietitians right now. I don't know why. Naturopaths. It's a wide variety of practitioners that take this course. And the goal that we all share in the class is to up-level our lab interpretation skills to become the best in the industry at interpreting labs, and to get, probably even more importantly, patient communication and program design skills. Program design is a personal focus of mine for a long time, meaning like how are we going to design a program? How are you going to get therapeutic dosages of every product? How are you going to get the patient enrolled so they do all of this? How are you going to make it affordable, clinically effective, and lab-based? And that's a complicated equation. Just sequencing programs. You might know the right programs to do, but get them out of sequence, and it's not going to work very well. So there's a lot to learn, and we do this as a group. It's at least a 3 to 5 hour commitment per week. Some people put in more time than that, but that's the minimum amount. And by the end of the year, you will be very comfortable interpreting adrenal and female hormone and GI and metabolomics related tests. Yeah. And I'm just thinking because there's a whole group of people that just finished this year, this month, a group that started last year in September, and I'm talking to all of them now. It's a pretty profound experience for people. Let me just put it that way. People are really, it's life-changing for the practitioners that sign up for this course. Okay, it's a very small group that we take every time. All right, so let's take a look here. Now, this is a neutral valve from Genova. I've also got a copy of a omics test from DSL, or Diagnostic Solutions Lab. These are the two main lab companies that I know that do amino acid testing. Okay, the formatting is almost identical for what we're talking about because it's such a straightforward lab. So we're skipping huge parts of this test, obviously, right? Because we're just focusing on amino acids today. Let's see if I can find the right part here. Sorry, let's scoot around. Here we go. Now remember, you can get urinary amino acids done also, but that's not what we're talking about. It's much preferred to do plasma amino acids, okay? So it involves a blood draw. And you can see them laid out here. It's not that complicated. If the amino acid is low, there's something that's causing that to happen. If the amino acid's high, either they're over-consuming that amino acid in supplement form or they're not absorbing it. It's not going into their cells very well, which can also, often means there's a B vitamin deficiency of some kind. Now remember, out of these amino acids, let's say on this particular case, arginine, histidine, isoleucine, and leucine are low. I'm going to write that down because I'm never going to remember that. Arginine, histidine, isoleucine, and leucine. So for each of these amino acids, you do need to spend, probably a couple weeks or a couple months studying that amino acid and what it does and why it's so important in terms of its non-protein functions, because remember, those are all different. And let's go back though to our list here. And this is the list of the 20 that are required for protein synthesis. So let's just look at this patient had low arginine. And this is, there's arginine. It's on the list. Histidine. It's on the list. Isoleucine. Oh, and here's leucine. Not every amino acid's, in this 20, obviously. And here's isoleucine. So for this particular patient, they're low in 4 of the 20 amino acids. 4 of the 20 amino acids. That means that there's 4 reasons why their protein synthesis is not working well. And remember, you take these 20 amino acids in about the same proportions, you string them together in strands in order to make protein. It's your DNA that's telling the amino acids how to arrange themselves. But none of that's going to be effective if there's 4 of these missing. It would be like if you imagine a bicycle chain that's, you're locking your bike up with, and 4 of the links are broken. It's not going to be a very effective chain. It's going to be useless, basically. Okay, you cannot synthesize proteins without all of these amino acids being present. And this explains why many of our patients have what seem like global problems, like they're tired and they're overweight and they have joint pain and they have this and they have that and they have this and they have that. Because if you can't synthesize proteins, you just can't repair and heal, and the systems throughout the body start to shut down. The treatment for this is about as simple as you can get. You provide the missing amino acids in a powder form, usually for 3 to 6 months. I like to do it for 6, then you stop, and then you retest, okay? Now, for each of these amino acids, you can also think about the other things that they do. So arginine, for example, is required for the urea cycle to eliminate ammonia from the body. It's also required for nitric oxide production. So that's the healing, stabilizing, repair, restorative stuff that hits your blood vessels and makes your blood vessels really healthy. Leucine is very strongly involved in muscle growth. What was the other one? And isoleucine as well. Here's our tryptophan. In this case, it's normal. And so even though some of these are a little on the obscure side, like you probably haven't heard too much about arginine, if it's on that list of one of the 20, it's an absolutely critical nutrient. They divide up amino acids also between mutually essential amino acids and non-essential, and that's really misleading in my mind. I know the essential ones you can only get from food, non-essentials you can synthesize, but the non-essentials, just to show you here, it makes when they say non-essential, it makes it seem like it's not that important. But let's just take a couple of examples of the non-essentials: glutamine, tyrosine, cysteine. Now let's look at our list again. Back here. What did I say? Glutamine. Yep, it's there. Cysteine. Yep, it's there. Tyrosine. So these are technically non-essential amino acids because you can synthesize them in the body, but they're one of the 20, each of these is one of the 20 that are required to build proteins. So in a sense, non-essential makes them seem like they're not important. They're extremely important. If they're low, you've got a major problem with protein synthesis. Okay, let's take another example here. Here on the omics test from Diagnostic Solutions. They also do amino acids. And make sure you ask for the plasma amino acids, remember? And here they have the, on the back page, they do a summary. Throughout the actual report, they'll list the individual ones. On the back page, they'll do a summary of the amino acids and let you know if they're high or low or what's going on there, okay? And let's look at another Nutraval. Here. So there's lots of other things on this test, obviously. We're just kind of looking at the important stuff here. So now, neutrally essential, non-essentials. Isoleucine, leucine, again low. Methionine, we talked a lot about methionine earlier, is also low. That's on our list of one of the 20s, one of the 20 amino acids. Proline, that's usually not supplemented directly, okay? And I'll show you another example of how to actually design programs, and then we'll get to questions. So let me just show you one program design thing here. So in general, with the amino acids, there's easy formulas that are free-form amino acids all in a powder. And there's a couple of different brands in most of the professional lines have some version of it. And I'll show you I'll try to show, a more general way of how you would do this versus a specific lab. And most of the time I'll do it with a powder because it's just easier. And this powder is often combined with a bunch of other things. So let's go here and here. So a typical free-form amino acid powder, 1 teaspoon 2 times a day away from food. That would be a standard dose of all the different aminos at one time. That is going to be equal to approximately 5 grams 2 times a day. Be careful when you're ordering these different products from different companies because they all have different size scoopers. So sometimes a scooper is 3 grams, sometimes it's 12 grams. So don't let the patient take 1 scoop 2 times a day. Let them know, hey, it's 1 teaspoon. Throw the scooper away if you're confused or they're confused, and just have them get a teaspoon like you would bake a cake with, right? A teaspoon of sugar or salt or whatever, and have them measure it with a teaspoon. But be careful of that because I've had patients who use the scooper, and sometimes if the scooper is like a 12-gram scooper and you're giving them 2 of those a day, that's going to not be the right dose. Okay? Now, this is often then combined, like in this example here, with other products as part of a mitochondrial repair program. Okay, so not most of the time, all of the time, what we're talking about today is integrated in with another program. So if we're doing mitochondrial repair, it would be free-form amino acids plus CoQ10, magnesium, carnitine, NAC, B complex, and a multi-pack. Okay. That's a super common way that I would do it. Now, if you want to get more granular, and let's say you have a case and there's low tryptophan, low tyrosine, and low glycine, then you can do a standard free-form amino acid powder, which will have a little bit of everything in it, as sort of a base, and then add to that some glycine. You can get glycine in a powder. A typical dose for a patient with low glycine would be 3 grams. Let's say you're doing that 2 times a day. So it's pretty easy. You have the patient do their free-form amino acid powder, a teaspoon, then they put in their 3 grams of glycine in there. And then if they're also low in tyrosine, you might have them do 1,000 milligrams of tyrosine 2 times a day. Again, they can just open up the capsules and dump it into this one drink, and they're low in tryptophan. So you give them, let's say, 500 milligrams of tryptophan 2 times a day. And this is all be in one drink. In fact, what I'll do a lot of times is I'll have the patient mix up the full day's worth. So it'll be 2 teaspoons of free-form amino acids. Let's see, this is wrong here. It's 3 grams total. So let's say 3 grams once a day. It'll be 3 grams of the glycine powder, 1,000, 2,000 of the tyrosine, 1,000 of the tryptophan. Mix that all together in a drink. Have them shake it up. Drink half of it in the morning, half of it later in the day, so they don't have to make the concoction twice a day. So you can make it pretty simple for patients. And rather than them taking 4 different types of capsules and all kinds of pills, that's their whole program right there. If you're going to use amino acids effectively, we're not talking about this too much today, but you also need some B complex in there in order to utilize them. So whatever your favorite B complex is, and make sure it has plenty of B6 because B6 is required to make all these work. So if you think about it integrated into a GI program or an adrenal program, or like I showed originally a mitochondrial program, it's not that bad. Even though you've got all these aminos going on, it's really just a drink, half in the morning, half in the late afternoon, and they're done with that whole part of the program for the day. Okay, I'll give you another example here. Let's say you don't want to use the free-form amino acids for some reason, and you're just going to go for the individual ones that are low. And I'm just trying to show protocols that I use on a regular basis that I know you're going to need to use once in a while yourselves. Let's say they're low in arginine and citrulline. That's a common one. So they're low in arginine. So you're going to do arginine, say 1,500 milligrams 2 times a day. And you're going to do citrulline because they're also low in citrulline. 1,500 milligrams 2 times a day. Obviously, it's a total of 3 grams of each of those. You have your B complex in there. Like we mentioned, you always need the Bs. And let's say the patient's also low in, what's a super common one that's kind of fun? Oh, like glutamine. And that's glutamine powder. And that would also be, let's say, at least 1,500 milligrams 2 times a day. So glutamine powders are faintly sweet. So if you have them do this in a drink form, they can do the glutamine powder with some arginine and citrulline in it, put it into a drink, drink it down. That's a lot easier than just trying to take a ton of pills, okay? And if you want to do it with capsules, you can too, but it ends up being a lot of capsules for people to swallow. If people can't handle the drinks or they taste so bad that they can't handle them, you could break this down into capsules, but it's just much more difficult. Okay, there are also some companies out there that'll make custom amino acid formulas for you if you send them the lab results. It's a little complicated to do it that way. I usually just try to make them up with the things that I have available to me from the general supplement companies. All right, so just going to wrap up and then we'll go to questions. General theory here, if the only thing you remember from this is this one fact, I'd be super happy for you and for myself have had a very productive 45 minutes, which is that there are 20 amino acids that are required to build proteins in the body. What proteins? All proteins except for collagen. If you're low in one of these, like glycine or tryptophan, the reason why you want to give that tryptophan is not because their serotonin might get better, but instead because it's required for them to build proteins that regulate immune function, proteins like hemoglobin, proteins like all the enzymes that run your metabolic systems. All of that is requires all 20 of these amino acids. So that's the main reason why we use amino acid therapy. And if you use free-form aminos, it's kind of like cheating a little bit. You don't have to get super specific. They're going to have all these aminos in it. You add in extra for the ones that are low, and you make a sort of a custom drink for people so that they're getting amino acid repletion for 3 to 6 months. Then you have them stop and retest, and you always want to include at least B vitamins in there and probably some kind of a multi so they can utilize the amino acids effectively. And usually they're taken on an empty stomach twice a day, typically upon awakening and in the mid to late afternoon. For a lot of patients, they're very energizing and I would say quite pleasant to take. You don't get a lot of complaints of people saying, these amino acids are, not good. Most people are like, oh, this is kind of, satisfying. Quick reminder, we have a mentorship class coming up, and I'm going to take a peek now at questions. So urinary amino acids, I've only learned how to interpret plasma. I don't know how to interpret the urine ones. When I was studying with Richard Lord, who is the scientist that developed amino acid testing, and I brought up the subject of urinary amino acids, he said, I won't teach you that. and the subject never came up again. So clearly he was saying, don't waste your time, just do plasma. I mean, really what he was saying was, if you're going to bother testing amino acids, do the best test, get the best data so you can make a difference there. Can you have amino acid stores that are adequate if you're a vegan? Yeah, absolutely yes. I mean, some people would say plant-based proteins are superior. I don't know. I'm an aspirational vegan. I did have ground beef for lunch, so I'm not a very good vegan, but I wish I was a vegan in a lot of ways. Let's see here. So for the 12-month program for Cheryl, we have all kinds of different practitioners. If you're a health coach or something like that, just set up a call with me and we can talk through if it's a good class for you, a good fit or not, okay? But we have had a lot of personal trainers and health coaches take the course, but it's better if you don't sign up for it until we talk in person and make sure it's gonna be a good fit. If most amino acids come back as low, you've got a big problem on your hands and you're gonna see some big changes when you get that person better, okay? Now, obviously, if there's a huge pattern of low amino acids, it could be that they're not eating enough protein, or it could be that they don't digest well. And with digestive problems and low amino acids, the first thing I would think about looking for would be H. pylori. Plasma amino acids, plasma amino acids, plasma amino acids. Let me say that again, because people keep asking. I only teach and only learned how to interpret amino acids in the blood. I don't even know what the urinary ones are for, to be honest. Richard wouldn't teach me that, and so I just figured, hey, I'm going to learn from the master. I'm just going to do what he tells me. I think it's because when you're a lab director and lab formulator and you've been doing amino acid testing since the 1970s, as Richard has, the idea that you would do an inferior test because it's more convenient is just sort of offensive, you know? Pretty offensive for him. It was the only time he ever said anything like that to me. We worked together for like 14 years. And he didn't put down urine amino acids. He said, I'm not going to teach you that. And it was just, we moved on. And he basically said, just do the plasma, kid, you know. Now here, Cheryl's asking a good question here. I was alluding to this earlier, but let me just show you how complicated this is. This is why you're going to all sign up for the mentorship later this evening. So I'm going to make up something here, but let's pretend that this patient has normal tryptophan, okay? Which I actually think they do. But now we're looking at the serotonin marker here, and we see that it's quite high, okay? So the tryptophan is normal. Can you give tryptophan to somebody with normal tryptophan and a big serotonin problem? That's a question. Who thinks yes? Who thinks no? Can you give tryptophan to a patient with normal tryptophan that has a big serotonin problem? Christine got it right. Out of all you people out there, there's hundreds of you, someone else has got to say something. Yes, okay. Analia got it right. Kathy's right. Cheryl's right. Okay, you're all coming in now. All right, you're paying attention. Yes. The answer is yes. If tryptophan is low and the serotonin markers are messed up, that's like extra bad luck. That's like really bad luck. I don't think this has ever happened to me, but that would be like if you're driving a car and you got a flat tire and you're trying to get off the freeway and you're like, I may not survive this. I'm going 80 miles an hour and I have to get all the way over to the far right of the freeway with his flat tire. What's going to happen? And then on the way over there, a second tire went flat and you're like, oh my gosh, not really good. So you can have both these problems at the same time. You could have a serotonin marker that's positive and low tryptophan. That just means the person's got an extra deep problem, okay? And this is, I think, one of the more important clinical nutrition concepts to get. I'm going to try to explain this, that You can give a person tryptophan because they're low in tryptophan. And why would you do that? What's the most important role of tryptophan? Protein synthesis. Doesn't matter what's happening with their serotonin. You don't care at all, right? And this person is not on an SSRI, by the way. Anne was super smart and saw that, but they're not actually. We had a conversation about this earlier in the week with the doctor whose case this is, which is even more interesting that their serotonin marker is so high and they're not on an SSRI. But then the person with low tryptophan, well, let me say it this way. If you have low tryptophan, you have to take it just to get your levels up to normal. But if you have a process in the body that requires more tryptophan, you can supplement with tryptophan even though the tryptophan levels are normal, because you're trying to get a specific metabolic process working. So let's think about, like, I think magnesium is an easy way to think about it. If you test low in magnesium, obviously you have to take magnesium. And almost all of our patients are low in magnesium. But let's say you get a patient who's got normal magnesium levels, but you want to try to lower their blood pressure using magnesium. You can still use magnesium, even though their magnesium levels are low, are normal. You can use magnesium to have an intended effect. So in clinical nutrition, sometimes you're using magnesium to lower blood pressure, irregardless of whether their magnesium levels are low or not. So in clinical nutrition, we're always wanting to make up for a deficiency. That's the minimum lowest bar. But you can also use, for example, tryptophan. If their tryptophan levels are normal and all their amino acids are normal, but they have a serotonin problem, you can still use tryptophan to help with their serotonin. So they're related but somewhat separate issues. And sometimes you have both. You have two flat tires, right? You got low tryptophan and you have serotonin markers, and you try to fix them both at the same time. Let's see here. If an amino acid is high, it generally means that it's circulating in the bloodstream and not getting into cells. If methionine is high, you want to look at the methylation pathway and see what's happening there. And this is where it comes in handy to know all these different pathways. Let's see. If we have any more questions here. I think we got through most of them. All right, I'm going to wrap it up for now. Okay, take care, everybody. Thank you all for coming out for the evening, and I hope you learned something about amino acids, and I look forward to talking to you in the next one.